So enzymes have some standard nomenclature that you'll need to know about, and then we'll talk about the major classes of enzymes that you'll need to be familiar with as well. So enzymes have the family name ending in ASE. Anytime you see this type of nomenclature, you should be thinking that this is an enzyme that's catalyzing a reaction. So there are some exceptions because this is the standardized nomenclature. Before the standardization, there were some enzymes that had previously been discovered and named, and so they kept their older nomenclature before this new one came to be common practice. So a lot of these were the protease enzymes that will mediate the breakdown of proteins, especially within the digestive system. So these things are things like papain and trypsin, which are going to be referred to by their older common names. So our modern systematic names always have two parts. So the first part is going to identify the substrate on which the enzyme operates. So we try to name the enzyme for its activity. The second part is the enzyme subclass name. So, and we'll see some examples of these on the next slide, and we'll actually get a feel for what different types of subclasses of enzymes are going to exist. And so these are actually the reaction types or the classifications of our enzymes. So for example, our pyruvate carboxylase is a ligase enzyme that acts on the substrate pyruvate to add a carboxyl group. So now you're thinking, ligase. I've heard about a ligase enzyme. We heard about the DNA ligase. So that is a different type of ligase. That's going to ligate to the backbone of the DNA molecule. It's going to take the phosphate and the sugar and link them together. So ultimately, a ligase is anything that brings two things together and kind of hooks them together. So with the pyruvate carboxylase, the pyruvate molecule is being linked to the carboxyl group. And so that enzyme is acting as a ligase because it brings these two things together and hooks them together. So overall then, our latest systematic nomenclature system is known as the International Enzyme Commission System, or the IEC, is going to be based upon the type of reaction that's being catalyzed. So there's 6 broad groups of enzymes within this system that are shown in this table. So these represent overall families of different enzyme types, and you'll need to be familiar with these major classes of enzymes. So we've got our oxidoreductases. They're going to mediate oxidation reduction reactions, so the shuttling of electrons. Oxidoreductases are going to be very important for metabolism, or the formation of the ATP molecule. Transferases are going to transfer functional groups from one molecule to another. So they essentially transfer groups. We've already seen a lot of hydrolases. They're going to add or remove water molecules from a compound. So a lot of the enzymes that are going to mediate the formation or the breakdown of large biomolecules are hydrolases. So remember, when we put 2 amino acids together to form a peptide bond, we are going to release a water molecule. And so the enzyme, the ribosome, that's mediating that reaction falls into this class, the hydrolase class. Lyases are going to add or remove groups to form double bonds. So we saw carbon-carbon double bonds in Chemistry 105, the formation of the alkenes. So these would be mediated by enzymes that fall into the lyase class. Isomerases will transfer functional groups within a single molecule to form an isomer. So we know that isomers have the same molecular formula, but they have a different arrangement of the atoms in space. So enzymes can actually mediate the transfer of groups around and rearrange the molecule to form these isomeric structures. And then ligases bring 2 substrates together to form a new product. So 2 things building into a bigger thing. So now we'll see examples of these different reaction classes. And we'll see that enzymes can actually be characterized sometimes into more than one class. So we're going to see this first reaction is going to take our molecule of succinate plus a molecule of ubiquinone, and it's going to create a molecule of fumarate and ubiquinol. So if we look closely at the chemical structure, can you see what's going on here? What's different about the succinate and the fumarate molecule over here? Hopefully you can see that there's a presence of a double bond that gets formed here. So you've got formation of a double bond. So what class is that going to fall into? It should fall into our group of lyases, adding or removing groups to form double bonds. So we said we're talking about reductases here, but we can already see we must have a lyase reaction going on here. So we also have an oxidoreductase reaction going on. There's a shuttling of electrons to help form this double bond. So what do you notice changes about the ubiquinone structure? Hopefully you can see that there's a formation of the aromatic ring structure. So you've got another formation of a double bond in this molecule over here. And you've got the formation of the alcohol functional groups instead of, what is this called? What kind of functional group is that? Hopefully you all said ketone. So you've got the formation of the alcohol groups from the ketone structure. So if you'll remember our reactions, we can form by a redox reaction the formation of an alcohol from the ketone structure. And that's actually what we're doing when we build this molecule. So this molecule is going to lose electrons, and we actually can follow the loss of electrons and the gaining of electrons typically by the movement of the hydrogen molecules. So if we look at this carbon center over here, we'll see this carbon has one bond here and one bond here. So this carbon also has two hydrogens sticking off of it. They're just not shown in this drawing. And this carbon is the same, right? It's got one bond here, one bond here. So it must also have two carbons on it to be happy in the octet state. If we look at this molecule over here, now this carbon has one bond here, and now it's got two bonds over here. So it's got three total bonds. It only has one hydrogen now. It's lost that hydrogen. With the loss of the hydrogen, it also loses the electron that that hydrogen carries. This carbon is the same thing. So that hydrogen left as well, and it took with it its electron. Those hydrogens moved from this position on this molecule with their electrons, and they moved over to this ketone structure, and they attached with that oxygen. That pushed that double bond down into the ring structure, creating the aromatic core structure. And also the other hydrogen from the other position came and attacked that ketone structure. So you had the addition or the gain of the electrons, and the protons, the hydrogen, came with it in this molecule here. And this molecule lost the electrons. So this is an example then of an oxidoreductase reaction. And the enzyme that mediates this is the succinate dehydrogenase. And this enzyme is an oxidoreductase enzyme because it's taking the electrons from this molecule and donating them to this molecule over here. So it's mediating a reduction-oxidation reaction. So this also forms the double bond structure both in this molecule and in this molecule here. So this enzyme is an oxidoreductase, and it's also a lyase. So depending upon what feature is the more salient feature that you're trying to discuss with somebody, you might classify this into the oxidoreductase class and speak of it as the oxidoreductase enzyme if you're interested in the electron movement, or you may be interested in the formation of this alkene structure or the aromatic ring structure. And in that case, you may refer to this enzyme as a lyase. So ultimately, it's named as succinate dehydrogenase. So you see the ascending for the enzyme, and it's mediating reaction with succinate. So it's named for one of its substrates, and it's a dehydrogenase. So it is taking away hydrogens from the succinate. And that's exactly what this enzyme is doing. So this enzyme is named for its activity. So now we have the class of the transferases, and our example is a transketolase. And so this enzyme is transferring a ketone functional group from one molecule to another. So it's taking our ketone, you can see the ketone core structural group here, and it's going to take this functional group and it's going to transfer it, in this case, from our ketose to the aldose. So do you remember what a ketose and an aldose are? Hopefully you're remembering your sugar molecules. So our sugar molecules can either have a ketone core structure or the aldehyde core structure. If they have the aldehyde core structure, they're called an aldose. If they have the ketone core structure, they're called the ketose. So in this case, it's taking two sugar molecules and it's converting the aldose into a ketose, and it's converting the ketose into an aldose. And it's doing this by transferring the ketone functional group from one molecule to another. So this is known as the transketolase. So an example of our isomerase class is the triose phosphate isomerase. So remember, isomers are going to have the same molecular formula. They're just going to be oriented in space differently. And so this molecule has the same chemical formula. It's got this phosphate group, and then it's a 3-carbon molecule containing 2 oxygens, and it contains what, 5 hydrogens? So we can see that both of these core structures are the same. We've got 2, 3, 4, 5 hydrogens, 2 oxygens, 3 carbons, and our phosphate group. So, but in this case, they're oriented in space differently. You can see this has the ketone core structure. This one has the aldehyde core structure. And so we've got different orientations of the molecules in space. These are isomers of one another. So our dihydroxyacetone phosphate is being converted to the glyceraldehyde 3-phosphate, going from the ketone core structure to the aldehyde core structure. So, and then the ligases are going to be any enzyme that combines 2 substrates to make a new product. So we've already seen our DNA ligases doing this. It's taking the 2 strands of the DNA, joining them together into a larger strand of DNA. And we have another example then here that's called the aldolase. And so it's going to take an aldol and it's going to convert it into a ketose. So it takes our aldose, combines it with our dihydroxyacetone phosphate, and it's going to form the ketose core structure. So you're taking these 2 things, combining them together into a bigger unit. The hydrolases we're already familiar with. If we take our peptide bond, shown here, what type of bond is this? Our peptide bond? A little bit of review from the first part of term. Hopefully you all said this is the amide functional group. So if you've forgotten your functional groups, you should go back and review these. You still need to know them. So our amide functional group is going to get broken down with the presence of water. Remember, water can be added back to this bond so that you can recover the carboxylic acid functional group and the amine functional group of the downstream amino acid. So you recover the carboxylic acid and the amine functional group. So anytime you see water going into the reaction, or if water gets produced by the reaction, it's going to fall into this classification of the hydrolases. So protease enzymes are going to break down proteins through this process of hydrolysis. So this is an example of the chymotrypsin enzyme. It's a protease enzyme. You can see the polypeptide is going to enter into the active site of the enzyme. So this is a very nice diagram. Our polypeptide substrate then is going to be the amide bond. So we're going to want to break this apart. And you can see that there's a nice hydrophobic pocket that recognizes, do you recognize this side chain of the amino acid? Hopefully it looks familiar. This is the phenylalanine side chain, our aromatic ring. So this is one of our nonpolar aromatic side chains. It fits into a very hydrophobic pocket in this enzyme. So it's one of the docking features that helps this enzyme orient this substrate in the correct orientation. The aspartic acid, histidine, and serine in the binding pocket of the enzyme are going to help mediate the cleavage of the polypeptide bond. So you can see this enzyme actually forms a covalent link, an intermediate with the enzyme. So we said they sometimes could do that. And when it does this, it causes that peptide bond to break. When that happens, this portion of the molecule is now free, so you've freed this peptide. And then the water molecule actually has to add in here. So the hydrolysis actually is going to come into play here. And it's going to add back, and it's going to release that serine, which has that alcohol functional group. So see, that hydrogen went with the other peptide, so it's got to be regained from the water molecule. So the H goes back to the enzyme, and the OH is going to go up to the carboxylic acid functional group when this peptide portion gets released. So we'll stop this section here. Next time we'll talk about the effects of different substrate concentrations or pH or temperature that can have effects on the reaction rates.